Hello, welcome everyone. Thank you so much for joining us for another Museum Week program. So my name is Kerry Woke and I'm an educator at the museum. So normally I'm downtown planning the museum's big events like Bugfest, but today I'm here in my living room. So for Museum Week, we are focusing on the museum's research and collections section. As a collections-based museum, the Museum of Natural Sciences has thousands of specimens preserved to further scientific research. So today, we are going to focus on our ornithology collection and ornithological research. So of course, ornithology is the study of birds, and we have 27,000 bird specimens in our collection. So we're going to talk to John Gerwin and Brian O'Shea about the bird collection and their ornithology research. So John is the research curator of ornithology here at the museum and has done some fantastic research on southeastern birds, such as the Swainson's warbler and the painted bunting. Brian is the collections manager of ornithology, and his research is mostly focused on birds in the Guiana Shield region of South America. So welcome, John and Brian.
Thanks. Welcome, everybody.
Hey. So John and Brian, tell us about your respective research. I know you do work here in the Southeast, but you also travel to South and Central America to do research, correct?
Yeah, I've had a pretty good deal, I think, being able to travel to 14 different countries for some of the work that we do. And so it's pretty exciting, both in North Carolina and the Southeast, as well as some of the other tropical countries that we've worked in to study migratory birds. Anyway, it's been very exciting. We also do a lot of education and outreach as part of that. I think one of the coolest things that Brian and I get to do, he can comment more, is when we work out of the country, we work with local students and we provide training.
Yeah, and I work mainly in the Guianas of South America. That's northeastern South America, sort of east of Venezuela, pretty wild and remote part of the world. I've made two main research interests. One is the community ecology of understory rainforest birds, so looking at differences in species composition, individual condition, and just various aspects of their community dynamics and age distributions over time. And the other main component of my research is more socially oriented and in conservation, really. I work with Indigenous peoples in the southern part of Guyana, working to basically create conservation areas centered around a few endangered species that live down there.
And mostly what I've been doing out in the field when I do stuff is with putting little radio transmitters or other types of tracking devices on migratory birds to study their movements, both locally, like how they're using the space while they're breeding. And we've been involved in a few studies to show how they move across the Western Hemisphere, for example.
So one of my favorite stories about the Swainsons is how you put the transmitter on them. So can you tell us a little bit what you devised to make that work on such a small bird?
Well, there's some folks some colleagues we know came up with a little technique of a figure-8 harness, which is sort of like putting your underwear on with just the spandex. And it is thread that we use surgical thread because it rots over time. In case we can't catch the bird again, the thread will rot and the transmitter will fall off. It's a small bird like that. It's half a gram, and we're using 0.5 gram transmitters. They're pretty tiny, and we loop it around one leg and then have to loop it around the other and tie a little knot over the bird's back, so it sits on the back like a little backpack. But again, it's sort of like putting your pants on, and they can't get it off once it's a little bit tight. But it's a little bit flexible thread, so they can still feel comfortable. Yeah.
So that's so fun. So can you guys tell us a little bit about your craziest adventures as research biologists?
Brian, you can start with one.
Yeah, I think there's a lot. I was thinking about this the other day, and it came back to a memory I have of about 8 years ago. I was on a survey expedition in southern Suriname, very, very far southern border, near the border with Brazil. very remote area that we had to use helicopters to get into. And not only do we have to use helicopters, but we had to send an advance team up the river 10 days beforehand to actually cut a helipad out of the forest because we wanted to go to a particular spot. And there was nowhere to land the helicopter, so they had to go in there and cut away enough trees that we could put the helicopter down. And once we all got into the camp, we had a herpetologist with us who generally played a little fast and loose with things. And when he was dropped off, the helicopter pilot wandered into camp just to get some lunch, and they had the snake in the camp. And it was a really cool looking snake. And the pilot asked if he could hold it. And the herpetologist said, sure, not knowing what the snake was at all, not knowing anything about its toxicity or anything like that. Well, it bit the guy. And suddenly we had our helicopter pilot laid up in camp, having had some kind of reaction to this bite. We had no idea if we could get the helicopter out. And it turned out to be fortunate that he did recover, because a few days after that, our entire camp flooded and we had to evacuate. So good thing our helicopter pilot survived through that.
Did you ever figure out what it was?
We know what kind of snake it was, yeah. And it was a mildly venomous snake, with a rear fang mildly venomous snake that this guy apparently just didn't realize was venomous when he let the guy hold it. So those kinds of things happen in the jungle.
Well,
Awesome. So wait, hold on. Before I go, so I've got a couple questions already. So Linda wants to know, Brian, can you share an example of one of the conservation projects you work on in the indigenous communities in South America? Yeah, one of the major projects I've been working with involves a bird called the red siskin, which is a small red and black finch. It's extremely endangered. It was trapped relentlessly, starting really in the 1800s. And it's basically been wiped out from its native range, which is mostly in Venezuela and in parts of Colombia. About 20 years ago, a population was discovered in Guyana. And it turns out this population is actually quite a bit larger than the other populations that are in the historical range. So the area where it's found is pretty much entirely indigenous titled land. So we have been, there's a grassroots NGO based in that region with whom I've been working quite a bit to not only survey the siskin and find out more about its biology, but also to generate excitement about the siskin among the local communities, promote tourism based around the siskin, because now a lot of birdwatchers go to Guyana and they go to that area just to see these red siskins because it's the easiest place in the world to see them. So we've been continuing to expand that. We're working with the BirdLife people in the UK to get this place designated as an important bird area and hopefully get some more formal protection for it. Very cool. So is the red siskin related to our pine siskin? Yes, same genus. Cool. Awesome. In fact, it looks a lot like a pine siskin, except it's red. So. Awesome. Okay, so John, I've got two questions for you already. So Hugo wants to know is if the little underpants transmitter, does that affect them in any way when they try to fly?
Well, it better not, or it doesn't work to study the bird. So, a lot of people look into whether or not what those effects are. So this type of work is regulated, as you might expect. So we have to get federal permits to do it. And people have done a lot of work with captive birds to figure out what the limits are. And then you kind of back off a little bit. So most people are restricted to 3% of a bird's body weight. After time, you can go up to 5%. So I've done enough now. I have permission to use up to 5% of a bird's body weight. What we have to do when we're doing a study, we have to get all the weight data we can get from people for the whole year of a bird, just in case. I might be doing a project in June, but if we don't get the transmitter off that bird, we don't want to stress it if it's still wearing it, say, in September. So we look at all the weight data we can get. And that's the neat thing about the federal agency, that it gets all the data from researchers when we do live trapping and release. All of our data is submitted to this federal agency. So all I have to do is ask them, send me all the weight data for Swainson's warbler. And I get it all by the month or by the date. And I just graph it and I look for the lowest point. And then I make sure that whatever transmitter weight we use doesn't exceed 5% of that lowest point. So we do that with the birds. Now, when we're tying it on, if it's just a millimeter too loose, they escape. they know, they pry on it, they preen their feathers, and that thing slides right off. And we've had that happen a lot at the beginning because we didn't want to go too tight. If we tie it a millimeter too tight, they actually can't fly. You go to release them and they just jump to the ground and start running. So then you have to catch them and take it off because it's got a piece of glue on it. And so then you have to, we always work with, half a dozen transmitters that are already partially tied. And in case that happens, we just cut the thing off, grab another one and put it on. And then we just have to make sure it's not too tight and not too loose. You learn a little bit of trial and error. Like there's a length you can figure out, but within that length, every bird, is a little different. One bird is 15 gram, one is 16 and a half. And even those little differences make a difference for, like, again, just 1 millimeter off and that bird's going to escape. So we're very careful. And then after that, part of what we're doing is studying the bird's behavior. If we see anything anomalous, then that's a problem. But in the birds that we've studied, we have not seen that. A lot of other people, do that work as well. In other words, are the birds still reproducing? Are they feeding young? Are they incubating? And they continue to do all that.
So that just, you've touched on this a little bit, but Kim asks, so do crows, like the kind of the smarter birds, do they try to take off the tracking devices? And if so, do they succeed in getting them off? And have any of these efforts been documented on film or video?
Well, all the birds will try to get the thing off, because even when we put these little rings on their feet, we call it bird banding or bird ringing. So the little bands, the skin on the bird's foot it's essentially, well, it's like our skin, except they don't have really many nerves at that point on their feet, but they still don't like it. They look down and see these little rings and they try to pry them off. The same with something that's tied around, its waist. Essentially, birds have a different kind of waist, but it's fused with their lower back, but it's still there. They feel it, so they're always trying to get it off. And so again, sometimes they do, sometimes they don't. I'm not aware of any actual work with crows. We've actually talked about trying to do that with one of our other researchers here at the museum. He has access to some new tags that could be used on something like a crow, but I'm not aware. People have tagged them with wing tags. They've tagged them with these foot bands, leg bands, but I'm not aware of any telemetry work, is what we call it, radio telemetry. It's probably been done. I'm just not aware. But I would say that, yeah, if anybody can escape, it's a crow.
Right. Okay, awesome. So let's, all right. So John, you didn't tell us about your craziest adventure.
Well, I have several, like Brian said. Actually, I have one about a slightly venomous snake that I caught and didn't know it, but I got away with it. I would say that probably the real story is two that are related. 2 times working in other countries, we were interrupted by local politics. And probably the most severe was in Haiti, when we were there and we were supposed to work in 2 different mountain ranges, and it'll have to do with, maybe a bird story I'll talk about in a minute, is the black-eyed petrel, as well as colleagues studying a little thrush, a songbird that migrates there. And it was right during the election in 2006, which was supposed to take place in 2005, and it kept getting backed up and backed up. And Haiti is kind of a tumultuous place politically and weather-wise, climate. So wouldn't you know, it got delayed, and it happened like just 2 weeks before we went in February. And then they didn't make a decision, and everybody got naturally upset, and the place just imploded. So the UN was there, they were all over the place, and we spent a week in the field. And when we got back to the capital, we were supposed to go to another national park. They wouldn't let us. They said there were all these fires on the roadways, and what they were doing is kidnapping foreigners, and they were ransoming them for $10,000. And it was actually working really well for them. They were kidnapping a lot of journalists and a lot of other people, and they were getting a lot of money that way. And I had actually put 10,000 in my bank account before I left, because we were worried that might happen. And so my wife had permission to send the money. But what they did was they just said, you cannot go. American Airlines canceled their flights, and so we had to get a special small plane over to the Dominican Republic to get out. Like, everybody was caught, trapped in Haiti. It just was pandemonium. but we were working with a fellow who was connected, and he got us onto a plane over to the Dominican. So we made our escape. Anyway, stuff like that happened.
It sounds very exciting. So my next question is, can you tell us what has been, until now, because you guys are still researching, your favorite or what you consider to be your most important discovery? So if those are different, tell us both, please.
Well, I've been talking a lot, so I'll let, Brian, you want to say something about this?
I get excited about a lot of things, but I can say that, I wouldn't necessarily characterize my discoveries as something that's this big whiz bang, huge thing. What I really enjoy about doing the research that I do is the chance to get to know places and even individual birds very well. So I think that spending a lot of time in the field and being able to follow individual birds through the course of their life, this is, either in South America at my long-term study site, or even for the project that we did in North Carolina on painted bunnings that we did for several years a few years ago. We were going down and catching birds along the coast, and we were color banding them, so we knew them as individuals. And year after year, you go back and you see these birds, and you actually remember them. And it's really neat to be able to see them, to be able to watch their plumage transition from immature plumage to adult plumage, to look at its condition from year to year. Maybe you catch the bird in one area where you hadn't caught it before, so you're documenting and seeing in real time this movement that this bird is doing from say one territory to another, or one habitat to another. So all of these little things are really, really interesting to me, and they help you form a story. And I think that really it's not so much a single discovery as much as it is that your observations accumulate and allow you to kind of form this narrative and think about how things are working in the natural world. And that is something that you can do anywhere if you pay enough attention and you spend enough time looking at these animals.
I think that's so great. So I have some color banded birds in my yard, and I only have one. It's been a couple years, but I only have one, and he's a chickadee. And I think he was caught as an immature, and this year he's feeding babies. So that's like that story, right? So he's caught as a youngster himself, and now he's feeding his own kids. So. All right, John, what about you?
Well, I will. Like Brian said, there's just so many things. I'll mention two things, because one is more like cultural and one is it would be on a scientific thing. But just quickly on the cultural thing. So because I speak Spanish fairly well and I've worked in these other countries training people, I made 15 trips to Nicaragua, where I've been working with birds and tourism, but also training some locals. And what I found was that these are guys that didn't even go to they had barely finished high school that I've been working with, and they're so excited about their natural resources, and they were so excited to have these Americans coming down and then training them on all sorts of things, and they just became such good tour guides, and they learned so much so quickly. We'd bring them books and they would just memorize a field guide or some other like natural history book about the nature of their country, or even the US, because they're migratory birds all over the coffee farms where we were working. I know it's just one of the most rewarding things to see. And along those lines, it kind of carries over to a scientific thing. I've been working with teenagers for a while, and there's a few of them that helping me on this project in the mountains to study a songbird called the hermit thrush. And it migrates, but not very far. They're mostly a boreal bird in the north, and they migrate into the southeast. So we've been doing radiotelemetry, seeing how they do what they do. We've been measuring vegetation up near Mount Mitchell, where we've had our study sites, seeing what kind of habitat they're in. They started with me in 2016. They were still in high school, and now they're in college. And now, recently, a company has come along with a type of tag, a GPS tag. Excuse me. It's a data recorder. So you put the tag on, the bird does what it does for up to a year, and you've got to catch the bird again and get the tag off. So we tried last year, we put out 3 birds. And about 2 weeks ago, they went back up to the mountains and they found one of the birds and they got the tag off. And it was just so cool to see that, because they could program it to record 8 points. And the bird went to Alabama. So it was really cool to see, but it didn't go right away. So it hung around for like into mid-October. and then it just took off for Alabama. And when it was in Alabama, it moved around a little bit. It was kind of interesting because Brian has noticed hermit thrushes doing that sort of thing from his surveys, where they show up, but they don't stay around for, say, the whole winter. And that's what this bird did, and where it was in Alabama. It moved a mile over here and then over there. We had set it to record a few points in the winter, and then it came back, and it went right back to the parkway where it had been in September. So after the breeding season, it went over to the parkway, and then it left, and then it went back. Then it flew to its little territory. So it's only 8 points, but it was really exciting, you know, for these kids now who they first caught that bird in 2016 when they were in high school and banded it. And then they caught it last year, put this tag, and now they caught it again this year. So like you say, there's this story behind this one bird, but to put together its whole life history, that this thing flew to south of Montgomery, Alabama, and then came back right to the same spot. It's just very powerful to see this technology develop as we've come along. And, you know, that's with live birds and all the studies that people do with collections too. All this new technology has just been amazing to see in my lifetime.
I think it really speaks to the conservation as well, because when you get to know these birds as like individuals and what they're actually individually doing, you feel it's so compelling. You feel like, you know, that we have to really take care of these animals, because, you know, it's one thing, oh yeah, they're just out there, you know, we don't know, you know, doing their thing, whatever, but now we know what their thing is, and you know every point in their journey, and they need conservation in all these different places in order to survive. So awesome. Well, I've got a couple more questions. So Hugo wants to know, the other day he read about the hooded pitohui. I don't know if I'm saying that right, but it's the first documented poisonous bird. And do you have any experience with them?
No, but that fellow who discovered it, he lived with me for a summer. He skinned birds for us way back around 1988 or 89. He was one of our first part-time helpers. He was working at the museum up in Cincinnati. That's where I grew up, just north of there. And I was at a meeting, and I ran into him. We were chatting. He said he was looking for a job. We had a little bit of money back then. Might have been in 1990, but anyway, he came down and he hung out where I was renting, and he stuffed birds for us for a summer, and then went on to great things and make that discovery. That's the only experience I have with that.
So cool.
So, yeah, that's from that's from, as I recall, New Guinea. So, yeah, I've not been over there.
That's awesome. So Abby wants to know if you guys have a favorite individual bird that you've observed for a long time.
Oh, man. That's a tough one.
Well, I can tell you one. You know, I do bird surveys regularly at Prairie Ridge, and in 2013 in the winter, an American bittern showed up at Prairie Ridge, and that was a pretty cool thing.
Yeah, I remember.
And, you know, the bittern was extremely tame. You know, this is a heron that you can usually not see too well, and they usually don't allow you to get very close. But this one was initially it allowed people to get within a few feet. The next winter, it showed up again, and it sort of became a celebrity. You know, it was like kind of the flagship bird of Prairie Ridge for about, I think it was 4 years in a row that it showed up. I think the last time it was seen was in 2017. So I really began to enjoy seeing that bird, and you know, wondering whether I was going to see it on my walks through there. You know, I kind of knew where it hung out, and I knew, you know, what it did, and so I was always kind of looking for it. It's hard to see this thing, right? And you know, one time it snowed at Prairie Ridge, and I saw its tracks in the snow. The ponds had frozen over, so it wandered up into the prairie, and presumably was hunting mice or something up there. So it was really neat to kind of see this bird and know it was around, but sometimes not be able to see it. It was a lot of fun.
He was my life bittern.
Really?
Yeah.
Well, I remembered one. So it was one of those Swainson's warblers, because we did telemetry on them for about 6 years. And we started with birds that had already been tagged for a few years. We knew they'd hang around. And there was this one bird. He had pink and a lime green color band, and then another silver one with a number, and then a black and white split one. I'll never forget the pink lime green, or light green, olive green actually. And I wanted to get data on these birds. Well, that bird, we put a radio on, and we try to get like 30 point locations on a bird. We go find them and take a GPS recording. That's how we estimate what their territory size is. So I remember before we got to about 20 points, and I'm sneaking up on this bird. I could never see him. I would go out to look for him, and the technicians would go to look, and he would always like chip and fly away before we would see him. So we were forever estimating what bush he was in, shrubs, but we never saw him. We would hear him sing, never see him. By the way, the transmitters beep, and it beeps at a frequency, and it's a radio, so we have a receiver and an antenna, and that's how we find the birds from about 300 meters away. So we knew he'd be singing, we knew where he was, but he would never let us see it. And I come up really close one time, and I thought, great, I'm going to see him. And then I realized I'm circling. That meant that the transmitter fell off or the bird was dead. So I'm looking and looking, and after about an hour, I found the transmitter on the ground. So the next year he was back. So I put the transmitter on, and the same thing, never saw him. But before I got to 30 points, I found the transmitter on the ground again. Third year, same thing. I couldn't believe it. I did him again. Fourth year, he's still there. Now he's 8 years old, at least. We caught him when he was an adult. So now he's in his 8th year. And he got out again. He got out of the harness again. He's the only bird that's ever done that. Got out 4 times, and we never got like the full 30 points on him. But he was always in the same area, so we kind of knew. We could piece it together, and we knew what he was doing. But still, nobody ever really saw him all those times, except in the net when we catch him. It was just an amazing thing. So I give him a lot of credit. In the 9th year when we caught him, he had a really bad like skin disease, and it was really interesting. At first, I thought I had injured him, and then as I peeled the feathers back and saw all this weird growth on his skin, I realized it was just old age, because he was now in his 9th. He could have been 10, 11, 12 years. The oldest Swainsons we ever had was about 10 and a half years. So anyway, it was just really wild to, again, put that individual story. But he escaped every time.
That's a really amazing story. And and it's amazing that these tiny little things can be so long-lived, right? And I mean, migration's so expensive, and everything. So it's really amazing.
Yeah.
Okay, so Mike wants to know, how do you recatch the birds that you're tracking?
They will respond. Since we're mostly doing males that respond to their song, we just simply keep playing their song. So we might be, the batteries are set. We ask the engineer, we can tell them how long we want the battery to go, and it affects some of the signaling, but we usually try to go from 4 to 5 weeks on a little transmitter. So once we're nearing the end of it, we just start doing the song, the playback, and then a lot of times we get half the birds that we're studying. Like, we'll get them back. We can take the transmitter off ourselves.
Very cool. So you kind of answered it, but Glenn wants to know if the tracking devices are limited by battery technology in terms of length or life of device, et cetera.
Yeah, and of course it depends on what device it is, because now you get the big ones that can be solar powered that they put on turtles and albatrosses and whales and things, and but now they're much smaller. And then the radio transmitters are built with a hearing aid battery, and the engineers that build them, we got to know one fairly well. He works on under a microscope putting the little wires together, but he can program it because there is this little crystal that's doing its beeping, but he can decide for you. Like, you tell him what you want, and he will tell you like how frequently it beeps. Do you want it 1 per second, or 1 per 1.5 second, or 2 second? And then if you want it to be extra loud or strong, it will use up more battery power. So you can have it beep where you can hear it further away, but you won't be able to do it for as long a time period. So and it's not 1-to-1, unfortunately. It would be nice if it were like a 1-to-1 relationship, but he said no, unfortunately it's not. If you want extra range, you might get half more range, but you'll lose 80% of your power, kind of thing. It's not a simple math, and it's not in our favor. So, but so it's always a balance. But yes, it is limited by batteries, but some of that stuff is getting better and better too. Lithium batteries are better.
All right. I've got another question. So I'm going to combine two questions because they're really similar. So Allie wants to know if there's any birds you'd specifically like to work with in the future. And then Hugo wants to know what your holy grail bird species is, like the bird you'd love to study, but maybe don't have the opportunity.
Brian, do you want to say anything, or do you want me to take that right away?
Well, as far as birds I'd like to work with, I'm really interested in parrots. Parrots are abundant in the Guianas, and there's a lot, just that I've seen in my, survey work there that suggests that these parrots undergo these large-scale movements, probably seasonally, that we probably know next to nothing about. And I would really be interested in, as this tracking technology improves, I would really love to do some tracking of parrots. I think that would be a really interesting thing to do. Parrots are also very important, of course, for tropical forest ecology. They're seed predators, they're seed dispersers, so they really do influence a lot of what goes on in tropical forests. So parrots are really a high priority for me. That's really what I would love to do. As far as the bird that is sort of a holy grail, I don't really have one, I would say. There's certainly some species that I would love to see in the world because I've really only been in South America. I've never really gone anywhere else. So there's a lot of things I would like to see and just to experience being near, but I don't really have any real holy grail like that.
Well, Kerry, I think I would, if I could study a bird right now, would be the Carolina wren, if I had a lot of money. And the reason for that is that they occur throughout the Southeast, through the Gulf, and there are populations that go all the way to northern Nicaragua. It's probably a different species, and most people consider them, by the time you get, say, to Guatemala and on down, that it's really not a, quote, Carolina wren. But I don't think it's ever really been studied much. And the wren is an urban, it's a forest bird. It's really a forest bird, but it has adapted very well to an urban setting. And so I think that there are still questions that one could ask and look to answer about its biology and its adaptability. And actually, I don't think very many studies have been done of its, quote, natural habitat in the forest. It so quickly adapted to an urban lifestyle, and most of the work's been done in urban stuff. There's been some interesting work done with vocalizations. It has an amazing repertoire of vocalizations. There was a UNC professor did some work on them, but I think he, just got started, so to speak, and he's retired now. There's a lot of interesting things about bird vocalizations, and I think the wren, because of its habitat usage, two very different ones, its vocalizations and its geographic distribution, and it doesn't migrate, so there's some likely genetic structure going on out there. I think you could have a lot of different people involved and a lot of different levels, all the way, from neighborhoods or kids on up to the, the researcher at the university or our museum, coordinating a big kind of group study. And I think it would be a fascinating bird to do that with. So I have fantasized about that for years.
Yeah, it's such a ubiquitous bird that I always wonder like what these animals were like before, everything was so developed. Okay, so let's talk a little bit about the collection at the museum. So as I said, we've got over 20,000 bird specimens. So what makes our collection special or unique as compared to other museums?
This is where, if it's okay, I will shift over and just show, some of the slides. I'm going to share the screen here, and Brian and I will just kind of go back and forth with, see if it comes up. There we go. So you've, all right, so you've seen Brian and I both holding the same tropical bird, different places. And I'm going to see if it doesn't want to advance. Let me try this again. I don't know why it's not. I'm going to try it a different way.
So while John's working on that, I've got another question that, what's the biggest flying tropical bird?
Flying?
Yeah,
Flying.
flying.
Well, Andean condor probably would be the largest. I mean, tropical bird is, there are some seabirds that, of course, are found in the tropics and have, the longest wingspan. It depends on how you define large, right? It could be body size, could be body mass, could be wingspan. But in terms of just bulkiest, largest bird, Andean condor is up there. That's the one that comes to mind right away.
Very cool.
That's a good one. Yeah. Is it showing now?
Yep.
Yeah. Well, this is what the collections have. So we're just going to go through some different examples and show you. So these are the sorts of things that we curate in the collection. One of the kids that we work with was working on a project with me to do a blood chromosome thing, and she's showing some other youngsters how to take a blood sample. So that would fall under, for example, genetic resources. So bones are things that we do, and people borrow them for different reasons. Here are the folks at NC State University, and Julia used to be at the museum, did their work on some bones we have in the collection. Here's another one. Archaeology. I just wanted to mention, this is a pretty cool example of a study where one of our paleontology students, so from the paleontology lab at the museum, wanted to look at this thing called medullary bone. It only occurs in these in females that are, shows you the leg over here, mostly in the leg bones when a female is ready to lay eggs. So that's not very often during a given year. But it's not well known, but it's an interesting type of tissue. And they were looking at it to study how extant birds have it versus, extinct birds and dinosaurs. And so they wanted to first CT bones in our collection. So they did. She did CAT scans of all these bones. And then when she found them, she wanted to be able to cut some of them. So that's a destructive sampling thing that Brian and I discuss every study. And then we just ask ourselves, is this a good use of the collection? And in this case, we thought it really was, because they were studying the relationship of the tissue, the type, and the bird-dinosaur relationship. And they got some pretty cool results. And, some of our specimens did have this medullary tissue. So they got some pretty neat publications out of it. And they really advanced the understanding of this type of tissue. So that's a more like cutting edge thing. We still have eggs and nests. People still use these for different kinds of projects now and then. The eggs have been really good for getting the young people involved because they're always a lower priority, and we've had a number of donations over the years, and they sat basically uncurated. And when these guys came along and wanted to do work in the museum, so these guys came from the Waco Audubon's Young Naturalist Club, and Edward and some others came from our museum's junior curator program. Brian and I take them on and we put them to work on the simpler things that they can learn about collections, but also contribute. So they've been doing a lot of relabeling, rehousing, and then we shifted them over to work in the lab. But that's just one way. And then I'm gonna let Brian jump in here and talk about a little bit of what's down on B level.
So this is a picture of a couple pictures of our collections. We have about 4 rows of these archival specimen cases, and they're stacked, with drawers from top to bottom. And the bird's kind of laying there. You think of a stuffed bird as being a taxidermy mount, but that's not what we have. We have so many birds; they need to fit in a fairly tight space. So we prepare the specimens mostly as these study skins. These are birds that have been cut, and the bodies have been removed. Anything that can decompose or attract bugs or rot or whatever has been removed. The eyeballs are replaced with cotton. We do leave a few bones in. We leave most of the skull, and we leave the leg and wing bones in to provide some stability. We usually put a stick in the inside to keep the specimen together, and then we stuff it with cotton. And we have, I think at this point, we're up to about 27,000 specimens, and all orders of birds from penguins and other primitive lineages, all the way up through modern passerines, perching birds, sparrows and tanagers and things like that. We organize our collection by taxonomy, and all species are together. All related species are grouped together, and you go into our collection. You can just spend hours opening the drawers and looking at all of the different skins. John, you want to advance the slide one? So just to there's kind of a multiple layers of variation that we're looking for. On the one hand, we're looking at family level diversity. So we have a lot of different families that we've acquired, not only North Carolina specimens, but also more exotic things that we've acquired either through trades with other museums internationally or through the zoo, or I should say plural zoos. Zoos often donate their specimens to museums after they expire. And just to give you some idea of what kind of this variation can look like, just within one family, this is the hummingbird family. None of these hummingbirds are found here in North Carolina. Hummingbirds are mostly a neotropical family. There's about 350 species, and they're most diverse in the Andes of South America. So this is just a spattering of hummingbirds that you might find down there, including these guys up in the upper left, the sword-billed hummingbird, very spectacular bird, very large. Then you have in the kind of the center right here, the giant hummingbird, which is about the size of a large sparrow, that is found above tree line in the Andes. Hummingbirds, of course, are famous because their bill morphology reflects how they forage. So if you're interested in looking at foraging specializations, looking at broad series of specimens of birds of different families and different ecologies can tell you a lot about how these birds have evolved to exploit different niches. Can you advance them?
This one just shows, so this is one species, and this is the sort of thing that, again, that we can look at a different level, which is down within a species, but is there variation within? So people borrow our specimens, or we might be part of a project that we do it ourselves. And here's a case where these sparrows, and they're in coastal North Carolina, and you can see some are from the mid-50s, some the late 80s, but you can also see the handwriting. So over here, I've zoomed in on one to show you that these researchers were doing this project in about the mid-90s because there was more and more realization that these are coastal inhabiting what we call a group of salt marsh sparrows, and they've been declining quite a bit. And so people want to get a better handle on the population level diversity because it turns out they do vary. So that's why you see three names. You see this caudacutus, but over here you see they changed it to something called diversus. When you see this sort of thing, and I probably have it in one of the upcoming slides, is it's because they're breeding in a given location and only that location, and they start to evolve these different traits. So that's what people are studying. But you can also see that previously some folks were using ink. This is one of the little details where we don't really like to do that. Brian and I prefer pencil. That's what most people do. And you can see they had to make some changes down here, and it's harder when it's ink, but people put their initials because that becomes sort of a history of who did what determination. And so they were making a change, saying, no, you don't really have the caudacutus subspecies here in the town marsh of Beaufort County on that day. You had diversus. So interesting stuff like that. Well, there's a map just to show you that. So you see a bird that occurs like this is a saw-whet owl, and it's breeding, got some isolated population here, and this one actually goes into Western North Carolina. We've been starting to study them. And then you've got these birds here. So now people, would wonder, are these genetically different? Some preliminary work indicated that these birds are slightly different genetically from the rest. So, that has implications for conservation as well as evolution. So those are the reasons we do that sort of thing. Back to you, Brian.
Yeah, so just like John was saying, geographic variation is another layer of variation within a species, but there's also more basic variation just within one species that's related to age and sex. So here's a series of scarlet tanagers. This is a common bird, in North Carolina during the breeding season. Bird on the left is a juvenile, and then the one on the right is an adult male. And of course, they go through this transition of plumages, both through the year and as they age. To express these, well, really just to show how the non-breeding plumage gets replaced by this breeding plumage. And we like the scarlet tanager series because no two scarlet tanagers look alike. And it's also the same with summer tanager, which is a related species. Any two specimens in the drawer don't look the same, whether it be the intensity of red on an adult male or the pattern of splotching of red on these transitional birds. Or even just the intensity of color in the yellow spectrum on some of the female plumage birds. No two birds look alike, and so it's a lot of fun just to see what the level of variation is within a species. One of the uses of our collection one way collections are used quite a bit is by field guide illustrators. So if you're writing a field guide, you want to draw a scarlet tanager. What do you draw? How do you draw something that really shows what a scarlet tanager looks like in nature, so that people can go out and identify one? Well, you have to look at a museum series to see, the range of variation in scarlet tanagers. These are, and that might determine, how much space you allocate on a plate to drawing a scarlet tanager. Do you want to draw 4 of them, or do you only need to draw 1? So it's that kind of thing that, is really, really interesting from our perspective. We always enjoy seeing this kind of variation. I'll also point out that, preparing a bird specimen is a lot of work. And, it's not like people who study some taxonomic groups, you just go out, you put your net in the water, you dump a bunch of things in a jar, and you pour alcohol on it, and you're done. That's not the case with birds. Every bird specimen takes between 1 hour and 3 hours to prepare, depending on what kind of condition it's in, how large it is, how experienced the preparator is. So, one thing that I like to think about when I'm looking at a drawer full of specimens is just the amount of work that's gone into building this collection. And when your collection starts to grow and you start getting to 27,000 and 28,000, you really begin to realize the amount of work that's gone into building something like this.
So I have a question, guys, and this is, so Abby just finished reading The Feather Thief, and I'm sure you're both familiar with that.
Yeah.
So it just, to give folks who don't know, who haven't read it, it's about, it was the British Museum, right?
Yeah.
And it was a museum where, so somebody broke in to get these feathers from these bird specimens for fly tying. So there's this huge fly tying culture, and in some of these species are either are incredibly rare or endangered or extinct. And these were Alfred Russel Wallace specimens. Who Alfred Russel Wallace kind of came up with the theory of natural selection along with Darwin. And so these specimens were stolen out of the museum. So she wants to know if is security a concern when we are collecting and preserving our specimens?
Yes, it is. We anytime we get a request to look at specimens by a researcher, we evaluate the project, and we make appointments for researchers to come in and look. And we're usually we accompany the researcher throughout the whole process. So we're not overly concerned about it. In other words, it's never been a problem for us. It's certainly something to think about. But in general, the research community is they have a good sense of ethics about this. They understand, if you're doing research on specimens, you understand what specimens are, and you understand what they're for, and you understand the importance of them. So, we never go into hosting a researcher thinking that researcher is going to use our specimens for some kind of illicit purpose. We are always, I would say we're aware of it, but we're not particularly worried about it. And we do everything we can to stay around when the researcher is working in the collection. And that's mainly just to help them find things and just to make sure that everything is going smoothly for them.
We also have the security staff for the museum, and they are always checking the different floors. And we have some camera in operation. And I just want to point out that the thing that what happened at the British Museum was really a bit odd, because there was a major lapse in their own system. When there's been some investigative reporting done. There was a follow-up, and there was a fellow who did his own reporting just to try to figure out how did this happen. And what he uncovered was not very it didn't shine a good light on the British Museum security.
Right. Didn't it take them like weeks to notice that they even were stolen?
It took them weeks. The way the entry was done, it should have been found. The person who was monitoring, the camera monitor, was not really doing the job. In other words, people became complacent. So that's the thing that we have to always remind ourselves, is not be too complacent about any of this stuff, and that, because that's when you'll run into a problem. Right now, all the museums that we work with still have a functional security system where people are checking all the time.
I will say that incident at the British Museum was kind of a wake-up call for a lot of museums, though.
Yeah.
The British Museum is a huge museum. It's, in fact, the largest collection in the world. It's over 1 million specimens. So it's not entirely surprising they didn't notice right away. But still, I think a lot of people just kind of had become complacent about that kind of thing. And certainly it's on everybody's mind now that we know that kind of thing can happen.
I have a really good question. From Katie. She asks, what are some of the rules surrounding collecting nests or eggs or bird feathers?
Collecting nests and eggs or oh, right.
Yeah.
Right. Right. Oh, the rule is you can't technically. I mean, that's, yeah. I show a picture at the very end of our little slideshow that touches on the bird feather trade. And nests are a product of a bird's behavior. So that's an interesting thing. Once the nest is done, they're not going to reuse it. They don't. And in fact, we tell people to clean out your bird box. So, and really what you're collecting is vegetation. But of course, the intent of that law about not collecting nests was to not disturb the bird. I think most people know that. So that's not really a concern. When people find eggshells on the ground, of course, they want to pick them up. And nobody's really looking in your house for that sort of thing. I must know 1,000 people who pick up feathers all the time. It's just that because birds molt. That's what you're seeing right here. This bird is molting. It's changing clothes. And when they do that, they drop feathers on the ground. We have to tell people you can't do it. But of course, everybody does it because it's a feather that's just going to get washed into the sewer anyway. So, but that's just the rule. But Lord, we probably know 1,000 people with feathers all over their house or more.
But technically, it is illegal to keep feathers of any bird that's not a game bird taken in season.
Oh yeah.
Or an introduced species or something. Like pigeons, city pigeons. You can keep feathers of those, house sparrows, things like that. But any native migratory bird species, even non-migratory bird species, any native bird species in North America, you're not allowed to possess feathers of.
All right, thank you. All right, so you guys, I've got a few more questions, but I think why don't we keep going?
Yeah, this is just another example of what we have in the collection. We have a lot of spread wings that are separate from the skins because we were one of the first museums in the country to start doing this, and now we have probably over 5,000 of these that researchers and artists use. We also have a photographic library and when we're in the field. And I was just going to have Brian mention this other cool example of a use of these.
Yeah, recently we've had a graduate student from UNC Chapel Hill who has been basically 3D laser scanning our entire wing collection, which is a massive undertaking. It's thousands of wings that he's been doing. And he's been looking at wing shape, and what he's been finding is that the wing design is driven more by aerodynamics than by the need to structurally support the weight of the bird. So gliding birds have more efficient wings than birds that don't glide. And it's been a pretty interesting study, and it's been a lot of fun working with him. And he's one thing that he's done for us we really appreciated is that, like every museum has little curatorial problems. They have little missing numbers, maybe a specimen doesn't have a tag on it. Maybe information is really obviously wrong and it just kind of slipped through the cracks when the specimens were being cataloged. Maybe a specimen wasn't cataloged in the first place. And he's really helped us find a lot of these errors and help us to correct them. One other thing I want to say about our wing collection is that one real prominent application of this kind of specimens is that you can tell a lot about a bird's age by looking at particular feathers of the wing. And we spread these wings out because on a study skin, the wings are folded together. So you cannot see the details of the spread wing like this on a specimen. When the specimen's dry, it's brittle. You can't just pull the wing out because you'll break it. So it's really useful to be able to see just the condition of the flight feathers, which are the primaries and the secondaries, these long feathers toward the trailing edge of the wing. And also the coverts, the feathers that cover those feathers, the smaller ones. there has, a lot of the bird banders, especially those involved in the Monitoring Avian Productivity and Survivorship program by the Institute for Bird Populations, they are going out to these places and they're misnetting birds year after year during the breeding season. And their primary goal is to look at changes in demographics of these birds, so age distributions and reproductive success. And the primary way that they age these birds in the hand when they catch them to band them is by looking at their wings. And there are a couple of big manuals on how to do this, and those manuals were written using spread wing specimens like this, because you have the body data from the specimen tag that tells you everything you need to know about how old that bird is. You can separate known age, juvenile, hatch year birds from older adult birds, and you can look at differences in the plumage of the wing. And by doing that, you've been able to. These spread wing specimens have really contributed to our ability to be able to really get good age information on these birds, which has a lot of implications for habitat management and conservation, and just our general assessment of the trajectory of these species populations.
All right, I'm gonna see what's. We do sound files as well, but Ryan does more of that than.
Yeah, so the sound files are fun. This is something that I did a lot of even before I began working in museums. I was an avid sound recordist. I really love sound recordings. I looked at sound recordings for my dissertation. I have a lot of fun with this. But this is one point that I wanted to raise as well as we get to the end here, is that increasingly, we're looking at what we call whole specimen sampling. And so historically, of course, people would go out and they would collect a specimen and they would stuff it, and then they would put the skin in the museum, and that was that. But, as the techniques became available for preserving tissues and analyzing DNA, people began to collect tissue samples as well. Nowadays, we're looking at a lot more than that. We are trying to get recordings. We're trying to get, ecto and endoparasites, just about any kind of ancillary materials that we can get from a given specimen to maximize that specimen's utility for research we're trying to do. So it's really valuable to have sound recordings with specimens. There are some lineages of birds where the differences in vocalizations between populations can tell you a lot about genetic structure within a species. And so it's really useful to have this kind of extra information that goes along with the specimen. So we don't actually archive these recordings ourselves, but most of our stuff, in fact, really all of it, goes to Cornell. But the nice thing about that is that we can then link with Cornell, because everything's going online now, and all of these different databases are being integrated through various portals, that we can learn a lot about a specimen, even if the metadata are housed in a different place. So this is what the collections world is really going towards, and it's a really great thing to be able to do.
See what's next. Just another couple examples of just showing you like the variation that you see in a couple of species.
Yeah, a lot of people ask us, why do you have so many specimens? Why don't you just have one or two of each one? Well, the tanagers was a good example of the kind of variation that you see within a species. On the left here, we have screech owls. This is a common small owl throughout North Carolina. It has two main color morphs. One's gray, one's red. It's not too clearly visible, but the red ones are on the bottom here. And in our series right now, we have them separated by color morph. I don't know if there's any real geographic pattern to the variation. It's really just, you're either a red screech owl or you're a gray screech owl. But like the tanagers, if you look closely, no two screech owls look the same. On the right here, we have gray horned owls, and they display a lot more variation. Gray horned owls are found from the Arctic Circle all the way down to the southern tip of South America. And within that range, there's a vast amount of variation in these birds. But one of the coolest examples of variation that we have in our entire collection are these two great horned owls right here. These two are both from the state of Washington. They're just on opposite sides of the Cascade Mountain Range. So on the western side, where it's real humid and wet, you have a real nice illustration of Gloger's rule, which is that organisms in humid environments tend to be darker and have more melanins in their plumage. And so these great horned owls that live in that dark, damp Pacific Northwest rainforest are really dark. On the eastern side of the Cascades, it's kind of more of an open desert steppe sort of ecosystem, and great horned owls over there are extremely pale. And these two specimens were not taken very far apart geographically speaking, maybe a few hundred miles apart at most, but they look extremely different. And so it's really neat to be able to see that in our collection and be able to point out this really kind of extreme example of variation that you can see just within one species.
All right, guys, so we're just about out of time. So I don't know, is there something that, or can you tell us how are these collections helping with the conservation of species in birds? And what message do you want to leave us with why collections are just so vitally important for museums in the world?
You know, think of like one example, and you know, we had put stuff together because we never in here in the PowerPoint, not knowing how much time we'd have. But one thing we won't get to, but I'll just mention is one of the series of birds that we had was actually a rediscovery of something thought that would be extinct, and then they were found pretty commonly off the coast of North Carolina. But then more work was done, and this was the bird in Haiti that I was down trying to do some work with, the black-capped petrel. And then our former curator got some specimens and saw some slight differences, but in their facial pattern. And then, but he didn't really have time to follow up. He was doing other kind of survey work and even some work with plastics in their stomach. But now people are doing some satellite tracking of the birds. And sure enough, so they caught some out at sea. It's the first time that's ever been done, just 2 years ago. And, they got one of these birds that has a lighter face. It's a little different. And, they found it going to over into the Dominican side, into the Central Valley, not over in the mountains in Haiti. And that's what people were thinking, is that we had a researcher come up from UNC Wilmington and took some topaz samples from the specimens. And he found this one little genetic difference, just one. So it implied that there was something going on. And now with this satellite tracking, they're thinking, there is something going on, that there are two populations. But we really only knew about the one in Haiti. But now that's a conservation concern, is that their structure there's some genetic differences. Are they a different species? We argue about that still, but it becomes now an issue of trying to conserve both populations. And they think there's more now, that maybe over in Cuba, down in Jamaica, where they used to be, but now we're thinking maybe they're coming back over in the Lesser Antilles. And so it's spawned just this initial work that our former curator did in the 80s now has spawned all this research and all this effort going on, and training locals, training the Haitians and training Dominicans. And it's been taken over by some other people, and we're excited to see that happen. And it was a spin-off of using collections that way. And Brian, you may have another example, I'm sure.
Yeah, well, I just wanted to say that we showing this towhee here in this picture. This is one of the oldest specimens we have. We don't exactly know how old it is, but this collection date on this thing was sometime in the early mid 1800s, and it came from the Audubon collection. We don't know for sure that it was collected by John James Audubon, but it's a pretty good chance it was. This thing is, in the olden days, they used arsenic to preserve specimens. So there's arsenic soap to kind of rub on the inside of the skin, which is no doubt one reason it's so well preserved today. But, we preserve these things with an eye to the far future. These specimens are going to be around long after we're gone. And when people sometimes ask, do you really want another cardinal that hit my window? Do you really want, another, catbird or mockingbird? Well, yeah, we do, because there's a lot of news out there nowadays projecting into the future, and a lot of those scenarios look pretty bleak for a lot of birds. So there's no guarantee that what's common today is always going to be common, and we want to be able to preserve what's out there today so that people in the future can study what was there at this time. They're not going to be able to come back here and collect birds from this time. So we are always seeking to build a collection. We're always seeking to find new ways to use the specimens that we have, and as methods become more and more sophisticated for analyzing DNA and other materials that you can get from specimens, it's really important to have that continuity through time to be able to continue to add to these collections and to be able to put these animals away to be able to be used in the future.
Thank you, John and Brian, so much. This has been an incredible program. I do want to say there were a lot of questions we didn't get to today. So if you have them, and you'd like us to answer them, you can find Brian O'Shea, Kerry Woke, and John Gerwin all on the museum's website in the staff list. So, and you can send us any of us an email at any time, and we're more than happy to answer your questions. So thank you so much for participating in Museum Week this week. Tomorrow's the last day of Museum Week. We'll be talking to Brian Stewart, our research curator of herpetology, which is reptiles and amphibians. So please join us tomorrow at noon, and thank you so much. Goodbye.
Bye-bye.
Bye-bye.